Our next presenter is Sophie Alphonso. She's going to present her thesis on pollinator efficiency in novel ecosystems, a study of, help me with this.
Elateria, cardamom.
It's cardamom.
Yeah.
And the most effective pollinator. Her thesis advisor is John Vanden from EEB and Urban Environment, and her thesis reader is Chatra.
Chaturvedia.
Thank you.
Yeah.
I appreciate that. For EEB as well.
Yes. Okay, hi everyone. Thanks Jason for that introduction. Again, I'm Sophie and my thesis focuses on pollination efficiency in Mexico on the green cardamom plant. Again, pollination is an incredibly important ecosystem service that I'm sure you've all heard about, and it provides for humans in a multitude of ways, including for the global economy, for crop production, for biodiversity, and for biodiversity in general. And I hope this presentation helps shed some light on what pollination efficiency is and why it's important to study. So we're going to focus on some background first. So unfortunately, there has been a decrease in pollinators over the past 120 years or so. There's been about a 50% decrease in bee species in particular. There's a lot of reasons for this. Some of them include disease, pesticide exposure, and climate change. And one in particular that we'll be looking at today is habitat loss or landscape fragmentation. This can occur is what we're going to be talking about for agricultural management. So with changes in habitat or changes in global change, novel pollinators are increasing. The definition of novel pollinators are new or introduced pollinators in a given ecosystem or area. Novel pollinators have been shown to outcompete the traditional pollinators. This can disrupt regular plant-pollinator interaction in these areas. Studies have shown that plants are expected to adapt to the most efficient pollinators, even if these are not the traditional pollinators. They can do this in a lot of different ways. Some of them are increasing flower lifespan or size or increasing corolla tube length. These are just some of the examples here. So how exactly can we measure or discuss the most effective pollinator? So in 1970, Stebbins coined the most effective pollinator principle, and there's two components to this principle. They are the frequency of visitation and the single visit pollen deposition effectiveness. Overall, studying this is incredibly important because it can help us look at the effects of global change and how plant pollinator networks are being affected by this, and maybe if we need to enact some sort of conservation effort, and to what type and to what level. So our species of interest today is the green or true cardamom plant, also called the queen of spices for its flavoring and medicinal purposes. It is in the ginger family, and it's the second most expensive spice in the world after saffron. This makes its cultivation and production extremely sought after. It's native to India, but it is expanding. The production is expanding to parts of Mexico and Central America. So the actual growth of cardamom is incredibly interesting. It is often grown on coffee plantations, as cardamom grows best in partial shade conditions, and coffee can help with this. The good thing is there's no competition between coffee and cardamom for pollinators as they flower in different seasons. And as you can see from that photo right there, cardamom is an incredibly low-lying plant, so it also benefits from mulch or organic matter from shade trees above, another reason why it grows best in partial shade conditions. So cardamom has two unique characteristics that make it more attractive to certain pollinators. The first are those corolla tubes. So that is that tube, picture's kind of blurry, I apologize. Right at the base of that plant, it's really long, as you can see, and the nectar is housed at the bottom of that corolla tube. That's what the pollinators want, like that's the goal for them. The second characteristic is that labellum, which is the kind of petal-looking area. It acts as a landing pad for pollinators. So what pollinators do is they'll land on that labellum, and then they'll ultimately touch that anther, which is where they can collect pollen. The anther and the stigma are right kind of next to each other. And then pollinators will use their tongue to go down that corolla tube to grab the nectar. While doing so, they touch the anther and stigma, which is pollination. Here are some pictures of what it actually looks like. So these are all bees, and you can see they're using that labellum as a landing pad, and they're kind of, some of them are attempting to put their tongue down the corolla tube to grab that nectar, which is that sugar-rich nutrient source for them. And then, however, not all pollinators in the region have long enough tongues to do this legitimately. So nectar robbing is a common thing that occurs for cardamom plants. The definition is taking nectar without physically touching those reproductive organs. The photo to the left is an example of a bee doing so. You can kind of see what they do is they create an incision right at the end of that corolla to grab the nectar without touching those reproductive organs. This is kind of complex because if that bee was to land on that labellum, because the anther is so close in proximity, they would collect pollen, so they can participate in pollination, but they just don't get nectar in that legitimate way. So from that background, I have two main research questions. The first one is just wondering who are the most common visitors of cardamom, and are these species, or which species are the most efficient pollinators of cardamom? So for my methods, again, this work is really interesting because cardamom is native to India. However, its growth is expanding to parts of Mexico and Central America, which we're looking here in Mexico. And there's no research on pollination of cardamom in Mexico or Central America. Only the research occurs in India. So our field site is an organic shaded coffee plantation called Finca Orlanda in the Sonusco region, right on that Guatemalan border. Here are two photos of that plantation. There are two parts of our field collection. The first one is observational. This is previously collected data from 2017 and 2019. Basically, bees were observed for a period of 10 minutes. The species was recorded, and so was the foraging behavior of that species. So these foraging behaviors include nectar collection, pollen collection, or nectar robbing. Our second part of field collection was our stigma collection. Again, previously collected data from 2019. Basically, the cardamom flowers opened for a period of 24 hours. So before they were opened, the flowers were bagged, like the previous night. Once the flowers opened, they were unbagged, and then the first visitor was recorded, the species was recorded, and then the stigma was collected and preserved. This was in order to measure single visit efficiency of a single pollinator of a single visit. We went back to the lab, and this is a photo of a stigma pressed onto a slide. We used fluorescent microscopy to show all the pollen grains that this specific species left on cardamom. So the point of here was to determine how many pollen grains each species left per each visit. So those like green fluorescent circles you see, those are all cardamom pollen grains. So we worked at counting those, recording that number, and then that kind of shows us what species has as many cardamom pollen grains as they do. So our results, from our observational data, we got some pretty interesting results. So first, there was a total of 17 species that visited cardamom in total. That's a pretty good number. These species didn't just include bees, they included butterflies, flies, hummingbirds, moths, and wasps. However, almost 98% of these visitors were bees. And again, there was 12 bee species total out of those 17 total bee species. So we're going to be focusing specifically on 4 of those bee species that visited. The first 2 are Eulema and Euglossa. These are the legitimate pollinators of cardamom. You can kind of see from the photo how long their tongue is, and that means that they can reach down in that corolla and grab that nectar in that legitimate way, thus pollinating the plant. They're both orchid bees, and Eulema has more of that honeybee color, and Euglossa is green, metallic, shiny, really easy to spot. These are pretty large bees. However, when we look at visitation data across all the species that visited, we see that Eulema and Euglossa, our legitimate pollinators, didn't actually visit that much compared to the illegitimate pollinators. There is almost 1,400 data points, and Euglossa visited about 20 times, and Eulema about 76. So again, not huge compared to like the 2 bars on the right. So the 2 bars on the right are Scapta trigona mexicana and Trigona fulviventris. These are both illegitimate species of pollinators. Trigona was our most abundant at almost 600 visits out of that 1,400. So these are those 2 species. They are native stingless bees to this area of Mexico that we are studying. They have short tongues, so they can't pollinate legitimately like Eulema and Euglossa can. So they take part in robbing and pollen collection. Robbing is from that incision at the base of the corolla to grab that nectar, and pollen collection is when they just land in the labellum and touch the anther. So if you remember, the second part of that observational data was determining what the foraging behavior was of each species. So we had pollen collection, nectar collection, and nectar robbing. We are considering pollen collection and nectar collection as a whole, pollination as a whole, as they require touching of some sort of reproductive organs of the flowers, while nectar robbing, it doesn't do anything with pollination and is its own thing. So for the legitimate pollinators we see here, Euglossa and Eulema, 100% of the time contributed to pollination and never contributed to nectar robbing. But when we look at the illegitimate pollinators of Scapta trigona and trigona, we see that they both take part in nectar robbing, but as well as pollination. So Scapta trigona had about a third of the time participated in nectar robbing, and trigona, over 50% of the time, took place in nectar robbing. So our second part of our data was, as you remember, that lab analysis of counting those pollen grains. So basically, a lot of similar values here, but we have those Euglossa and Eulema, the legitimate pollinators, had the highest amount of average pollen deposition on those flowers. Trigona was the lowest, but they were the most abundant. So putting it all together to calculate that pollination most effective pollinator, we use this equation, VR times DR times R equals DT. VR is our average visitation rate, or number of times each species visited that flower. And then our DR is that average pollen deposition that we looked at the slide previous, that graph. A higher average pollen deposition is a good thing. Our R is that number of flowers with receptive stigmas. In our case, that value is always 1, as we're looking at one stigma with one flower per single visit. And we multiply all those together to get that pollen deposition effectiveness. That's what we're trying to find. A higher higher value of DT is a good thing, and that means that that pollinator is the most effective. So doing all that math, we found that our most effective pollinator was Scapta trigona mexicana at DT equals 45. Followed by trigona, followed by Eulema, which is that our legitimate pollinator. Again, interesting results because our top 2 most efficient pollinators were our illegitimate pollinators, even though they also participated in nectar robbing. This kind of emphasizes how important it is to also look at visitation data, so the number of times each pollinator is visiting that plant. Okay, so just an overview, so we remember what's happening. Cardamom is visited by a wide variety of pollinators, which is a good thing, and that's what we were trying to find, if cardamom could survive and adapt to any pollinators that they had. Again, the most abundant was trigona, and the most efficient was Scapta trigona. And again, the most efficient pollinators also participated in nectar robbing, which was really an interesting phenomenon to find. So again, the research done here was in Mexico, and the only research available was from India, so we're going to compare that to the research done in India. The same genera was found in India, trigona, which was found to be the most effective pollinator in the native part of where this plant is native. They were also found to participate in pollination, as we found for Trigona fulviventris. There is one study that is not in India, from Papua New Guinea, that found 2 different species of bees to be the most, well, one to be the most efficient over the other, but those 2 species were not found to pollinate cardamom. In our case, Apis is the honeybee. So our limitations, one would be data. We need a lot more data to look at and make sure the results are adequate, especially for our legitimate pollinators of that pollen deposition. And then second would be to repeat locations. We are looking at an organic coffee farm. It would be interesting to see this work done on a conventional farm and kind of compare the results and see if they are consistent. So for my acknowledgements, I'd like to acknowledge my mentor, Chatura. I've been working with her since 2019, and her passion and her data was the reason that this project was possible, and I'm very appreciative of her. And I'd also like to thank John Vandermeer for being my advisor throughout this process. Okay, any questions? Yeah.
So after your findings, do you still repeat illegitimate pollination?
That's a good question. I would say yes, just because they don't, do that nectar collection in that legitimate way, and they do, we saw that there was robbing in 50% of the time. So if the legitimate pollinators and the illegitimate pollinators were to visit the same amount of times, that would be totally uneven, and those illegitimate pollinators would not be the most efficient. So I would say those names still are in place as of now.
So I have two kind of more methods questions for you.
Sure.
So, one is, I'm just curious, like, how you actually go about counting all of those pollen grains.
Yeah, maybe I can go back to it.
Question is, I wonder if you feel like your data might be a little bit different if the collection timing was different in terms of maybe time of day or time of year.
Mm-hmm. Yeah, definitely. So if I can go back to that photo. So obviously there's a lot of pollen grains going on here. We used a program called .goose, which you just have to like click each circle and it counts it totally for you, which made it a lot easier. And also we counted every single circle thing we found because each bag was unopened after the flower opened itself. It was only visited by one pollinator, so we're assuming that that's all cardamom flower grains. Coffee was not flowering at that point. And also wind pollination was probably pretty low because that flower was only opened for a short amount of time. And then remind me of your second question.
It was about timing, so whether or not the time of year or the time of the day of collection might influence something else.
Yeah, there's definitely a lot of things that could influence the results that we have, including like weather patterns or even like the amount of nesting sites that are near the area, if that's what made the abundance possible. I would say that. Mm-hmm.
Seems like you're off the hook.
Good work. Use the one I have. That's right. Running slightly early, actually. So we can delay. Forward. Press forward. So our next presenter is Samantha King. She will be presenting her thesis about insights into how litter manipulation alters soil extracellular enzyme activity in a northern temperate ecosystem. Her thesis advisor is Don Zach from the School of Environment and Sustainability and Program Environment, and her thesis reader is Amy Klassen from Thesis as well.
Awesome. Thank you, Jason. Yeah. So again, I'm Sam, and just to kind of simplify this down, I'm going to be talking about soil enzymes in the UMBS site up north in Pellston, Michigan. So just, if that works. What's the point of it? Where do I point it?
It's okay, this happened earlier too.
Oh, yes. It's not working right.
It's working on this, that's fine.
Connected to the wrong thing. Oh wait. I have to press resume maybe? Or wait. Oh wait, it's still doing this. There we go. There we go. Okay. Great. Okay, so kind of just a little bit about me and how I got into soil ecology. As a kid, I always loved being outside, being outdoors, and going to college, I realized that I wanted to learn how to protect these environments that I love so much. And particularly after taking a class called Woody Plants, I learned to identify hundreds of tree species and realized that I couldn't walk around anymore without wanting to understand all of these trees and all these environments around me. And so kind of the next step beyond that was to understand soils and how they operate and how they work. And so when I got into taking ecosystem ecology and soil ecology, and I talked to Don about doing a thesis and kind of doing research on this topic, climate change is the overarching area that is kind of in this field right now. And so we're looking at nutrient cycling and climate change and things like that. So kind of just an overview of the question that I was generally kind of trying to zoom in on was, how is climate change altering nutrient availability and nutrient cycling within ecosystems? And so when we think about climate change, we're thinking about increasing temperatures and greenhouse gases and things like that, and that's going to change our plants and plant material and plant litter. And so that's going to happen in positive and negative ways, just like climate change is affecting some ecosystems and actually increasing productivity in some, whereas others we're seeing drought and deforestation, which is obviously harming those ecosystems. And so in order to understand how this litter and these plants are being affected by climate change, we need to understand soil microbial communities, because they play a really important role in decomposing this plant litter and cycling it back into the environment. And so, yeah, just kind of in order to understand how climate change is altering nutrient cycling and things like that, we need to understand how this interaction is existing between plant litter and microbial communities. And so one study that it has existed for over 70 years now, that's a giant network across the US and internationally, is called the Detritus Input and Removal Network. And we have sites all across the US, but the site that I was looking at is the University of Michigan Biological Station. And most of these sites follow a similar treatment method. So we see there's a control, a double litter, which refers to leaves, no roots, no litter, no inputs, and kind of this AO layer, which is the organic material. And so I'll be focusing on the kind of left 5 out of those, but they all follow the same kind of framework. And when I was looking at the UMBS site, I wanted to kind of look into something that maybe hadn't, where was the knowledge gap when we were looking at this site compared to some of the other dirt sites. And this site hadn't had any research into enzyme activity yet, and so I decided that that was maybe an area that I wanted to venture into and kind of narrow the overall objective of these, the Detritus Input and Removal Network, of understanding how litter manipulation impacts soil organic matter, to then kind of zoom in and understand how quantity and quality of litter inputs affect activity of extracellular enzymes, which play a key role in kind of starting the process of litter decay. And so in order to understand how decay is changing, we need to understand these enzymes. So I did a little bit of field soil sampling as well as kind of some laboratory analysis. So as I mentioned before, my site was the UMBS dirt site. And that's up in Pellston, Michigan, in northern Michigan, right before you get to the bridge. And I used these 5 different treatments. I sampled all of them, and there's 3 replicates throughout the site. And I looked at control and then double litter. They take the litter that was on these no litter plots and they transfer it onto those plots. The no roots plots, they kind of excavate all the roots out, and then there's barriers below ground to make sure there's no roots in there. And then the no input plots kind of remove the above ground leaf litter and the below ground roots. And so as you can see there, I kind of used those soil cores to collect my samples. And then I also used litter fall data, so kind of data on the leaves that are falling into these plots. The most recent data was from 2015, so I used that data. And as you can see, here's just a couple pictures of the different plots. On the left, we have that double litter. On the right, the no litter. So we're taking leaves that fall onto that no litter plot, and we're transferring them onto that double litter to make that thicker layer of leaves. And then once I had all these samples, I transported them back to University of Michigan, where I did some laboratory analysis. And kind of for sake of understanding and a little bit of ease, I divide them into 2 categories, these enzymes that I looked at. Enzymes that decayed higher energy compounds, which were beta-glucosidase, cellobiohydrolase, and N-acetylglucosaminidase. So those are kind of like a mouthful, but the first 2 are really important in decaying cellulose and hemicellulose, which we tend to find in leaf litter. So when we think of these high-quality compounds and these high-quality litter, we're thinking of leaves usually. And then N-acetylglucosaminidase is responsible for decaying chitin, which is also a polysaccharide and a really high energy compound, but we tend to find that more in fungal root tips. And then the two oxidative enzymes that I looked at are responsible for decaying lower energy compounds. And these are phenol oxidase and peroxidase. And these lower energy compounds are things like lignin and organic matter, which we find below ground usually. And yeah, they just take a little bit more energy to decay, so they're a little bit lower energy and less ideal for the microbial community to use. And then once I kind of assayed all those enzymes, I looked at soil carbon content within all of my samples and did some statistical analysis to see if there was significant differences across the treatments and whether or not there was a correlation between litter fall and overall enzyme activity. So kind of leading into my results, I didn't get anything too surprising or out of the ordinary from what I expected. This graph on the left, we see these are the enzyme activities and the enzyme assays I did with the soil samples. So this is activity per gram soil. But we're going to kind of focus more on the right side, which is when I incorporated those carbon data and looked at activity per gram of carbon, and that gives me a better idea of kind of how microbial physiology might be changing in response to litter. And so when we look at these graphs, we can see that the general trends is that the double litter and the no roots were highest, had the highest activity across these enzymes that decay higher quality compounds. And that makes sense because we're having more leaves be added to these plots, or they're just still receiving leaves, whereas the lowest activity was in the no litter and the no input plots, where we're removing that source of high energy, we're removing that cellulose and hemicellulose, so we're not going to see as high of activity in those enzymes in those plots. And then we look at these enzymes that decay lower energy compounds, and there's less of a clear trend, but the highest activity is in these no input plots, where we've kind of removed all of these high energy sources, and so we're stuck with kind of the worst case, like just kind of the worst or the least ideal compounds left, which mean that we're going to see the highest activity of the enzymes that decay these compounds. Something that was a little bit surprising was that there was low rates of activity of these enzymes in no litter, which I didn't really expect. I expect them to be kind of similar to control or maybe even a little bit higher, but that was nothing super crazy. That was just kind of something notable. And then lastly, litter quality. I kind of ranked and I divided out species by their quality, and the black bar at the bottom is the high quality litter, all the way up to the low quality, which is that small white bar at the top. And this kind of just reinforces the idea that these plots were receiving really high quality litter. So there's a lot of cellulose and hemicellulose in these plots, and particularly these no roots and double litter plots were receiving, again, really high quality litter. So that makes sense when we think about the enzymes and the high enzyme activity of those, like beta-glucosidase, cellobiohydrolase, that decay these compounds. So just some kind of overarching key findings when we look at all these results together is that litter quality and quantity do impact the total enzyme activity and the composition of extracellular enzyme activity. And like I said, when we add high quality litter like leaves, we're going to see a jump in that enzyme activity of the enzymes that decay these compounds, whereas when we take those high quality sources away, we're going to see some more activity of those other oxidative enzymes. Another thing is, when we look at total enzyme activity, litter removal had significant decreased enzyme activity. And so that's something to think about in terms of climate change when we think about deforestation and removing leaf litter and things like that from our ecosystems, that we're going to see this decrease in enzyme activity, which will end up decreasing our kind of nutrient cycling within that ecosystem, most likely. And something else that was notable was that the no roots plot had similar overall activity to the control plot, which is not what I expected. And I think we can, based on the literature that's out there within the other dirt plots, this is probably due to the fact that the microbial community composition as a whole is shifting, so that may mean that it's shifting more towards fungi or away. I can't really tell from my data, but there is some sort of shift in the community composition happening. And then lastly, soil priming is probably occurring at these sites based on the fact that it's occurring at very similar sites across the other dirt treatments. And this is a phenomenon where when you add that extra, like an excess amount of that high quality litter, everything in the soil, all those microbes start to decay it more rapidly because there's more present, and that leads to actually the release of more carbon into the atmosphere because we're cycling through that. We're kind of increasing the respiration, increasing all of those processes. So that's actually not really a great thing when we think about that. So it's interesting to think about if we can see if that's happening more clearly at the biostation. So relating this kind of back to climate change and understanding shifts in enzyme activity, if we're seeing shifts in these enzymes, we're likely going to see shifts in nutrient cycling. And when we think about this in terms of the entire DIRT network and putting that in context, it just reinforces the importance of looking at a network like this where we have different ecosystems so we can understand when we manipulate this litter in all these ecosystems, what factors are important in all of these ecosystems and what are causing these changes. And so, yeah, the DIRT network really allows that. Things like precipitation may be really important, and we can tell that when we compare all of these different sites. And then lastly, limitations and some future research directions. There's a lot of places we can go. This study was kind of more of a baseline up at the biostation, but as we can see from a lot of the other DIRT research that's gone on with enzymes, there's a lot of places we can take this. We can look just at seasonal variations in activity, which can shift a lot based on, like I mentioned, like things like precipitation, which we know in Michigan that changes a lot from season to season. And I went in the summer, so if we go in the winter, things like that, it might be different. And that would give us just kind of a more robust data set and idea of what we're looking at with the enzymes. And then lastly, integrating data on carbon stores and soil organic matter and soil respiration rates and things like that can actually give us a better idea of whether or not things like soil priming are happening and what those shifts in the microbial community are. So I just wanted to thank everybody that has helped me get here today. Don is my thesis advisor, and he's just given me endless support and encouragement throughout all of this. And Amy, who is my reader, couldn't be here today, but she also just helped me up at the biostation with my data collection, as well as John up there. And it was my first independent research project, so couldn't have done it without them. And Rima, who's here today, helped me in the lab, taught me all of the analyses, and to my friends and family who came from all over the US today, thank you. Ooh, sorry. Yeah.
Really, you've done really interesting work. I'm wondering what this means for forest management, as you're trying to think through soil quality and health and enzymes. If you're trying to think through, I know it's a couple of steps away, but in terms of newsletter, forest management, what are some of the things that you have done?
I feel like just kind of understanding how manipulating this litter in general is affecting these forests and the cycling is really important. So when we look at maybe removing this litter and we think about deforestation and how that's going to impact activity, I think it just reinforces the idea that we need to maintain these ecosystems kind of as they are and allow these nutrients to fall into the flora as they normally do and not to remove any part of those. I know I was in a conversation talking, I think, to my dad about how we rake our leaves and things like that, just kind of at a at-home level sort of thing, and how it's better for the environment and nutrient cycling just to kind of leave those as they are. And so just, yeah, that would be my answer. Thank you. Yeah.
I was just wondering if you've done research on the biostation before, if this was your first time.
This is my first time. I actually was supposed to go up to take a class 2 years ago, and the pandemic hit, so I couldn't do that. So I went up in August for a week to do this data collection. Yeah, Don.
Nice job, Sam.
Thank you.
I'm wondering, you have some really interesting results, and I'm wondering if there's anything you want to follow up. What would be the logical next thing that you want to explore to pin down some of the implications that you have?
I think for me, when I looked at all of it, in the end, and looking at the research that exists at the other dirt plots, I would be really interested in just looking at the data on carbon stores and soil respiration and being able to integrate all of that to understand whether or not soil priming and what the shifts in the microbial community are, because I think that as much as this was really cool to do, I feel like there's a lot more that can be done. And so, yeah, there's I think being able to integrate all that together to get a better idea of the ecosystem as a whole and how it's operating would be the next step that I would like to take.
Thanks.
Yeah.
So you're interested in carbon content. Are you thinking that that carbon content in your dirt is contributing to greenhouse gas?
Yeah, so soils contain more than 3 times the amount of carbon than our atmosphere right now. So when we think about, they're not actively, they're mostly a sink of carbon right now, but depending on our land use change, like if we start to allow these gases like from the soil to be released, it could become a source, but right now I don't think it is. So yeah, it could depending on our land use practices, basically, so.